Hello, and thank you for joining us today. Welcome to our webinar entitled Targeting Kinases Involved in the Innate Immune Response. I'm Justin Brink, Director of Marketing here at Bellbrook Labs. Please note that if you have any questions during the webinar, you can type them into the chat box. We'll do our best to answer them during the live presentation. I'd like to introduce to you our presenter, Meera Kumar, Senior Scientist at Bellbrook. Meera has an amazing array of experience, from developing assays like this to our CGAS drug discovery program. She is well-versed in strategies for discovering, developing, and advancing small molecule inhibitors for enzyme targets. Thank you, and with that, here's Meera.
Hi, good morning, and welcome to the webinar titled Targeting Kinases in the Innate Immunity Pathway. Thank you, Justin, for giving the introduction. For those of you who are not familiar with Bellbrook Labs, we are a very small biotech company focused on developing tools for drug discovery and the high throughput screening platform. We've been here almost 20 years now, and in those 20 years, we have sold millions of reagents that have been validated by pharma, academia, and small biotech companies. So today's webinar focuses on kinases, and kinases in general requires no introduction. It's been really well pursued and studied target class. My talk will be divided into two segments. In the first half, I'm going to give an overview of the Transcreener technology, how it works, what are the different flavors, etc. And the second part of my talk, we would focus on kinases. And as a case study, we're going to briefly look at two kinases, what it entails to do assay development. And then we'll end with summary, conclusions. And as Justin said, if you have any questions, type in the dialog box or the chat box, and we would try to address it today. And if for some reason you're not able to get the answers today, please send us an email, or we will definitely follow up with answers to all those questions. Okay, thank you. Okay, sorry about that. So transcreener assays these assays are competitive immunoassay, and they're called immunoassay because we use an antibody, and this antibody was developed at Bellbrook Labs. It's a monoclonal antibody. So when we sell you a reagent, what you would get is an antibody and a tracer, and a tracer is nothing but the nucleotide that's conjugated to a fluorophore. And in the case of most of the transcreener assays, FT, FPI, or TR-FRET, we use a far-red tracer. And the reason we use a far-red tracer is to minimize any interference due to organic compounds. So our reagents are just very simple, an antibody and a tracer. And as you can see from the figure, it's a direct assay, meaning you produce the product from your enzyme reaction. In this particular, for this webinar, it's a kinase, so you would produce ADP, the adenosine diphosphate, and the antibody recognizes ADP, displaces the tracer, and we measure the signal. Now, there are so many kinase assays out there, and they're not bad, they're all good, but the biggest difference is our assay is direct assay. The coupled assays that are out there uses more than two, in some cases 3 enzymes that convert the ADP that's formed into a signal that can be measured. Now you can imagine when you're screening 1 million compounds, you will have to go back and deconvolute the assay to make sure it was not coupling enzyme 1 or the 2 or the reporter enzyme that was inhibiting your target, and it was the kinase. So it's a little convoluted in that approach, unlike our assay, which is just direct. So how does Transcreener assay work? So we start with an example of a kinase reaction. So a kinase would, you require an ATP, which is the donor molecule, and the acceptor molecule is any substrate. And we say any substrate because you could use peptide substrate, which is the most simplest substrate you could use, or you could use a full-length protein. You could even use substrates like DNA, nucleosomes, etc. So basically, it's a universal assay in which means you can use any substrate, any kinase, and then you just add ATP, and the kinase reaction would form ADP, and the phosphate will go to the acceptor substrate. So you have a phosphorylated product. Now, the way the assay works is so this is an example of the fluorescent polarization assay, the FP assay, where you have the antibody, which is in its native conformation, meaning nothing is attached to it, and you have the tracer. So at time 0, or when the kinase reaction has, let's assume either time 0 or the reaction has not proceeded at all, the antibody is bound to the tracer. Now, this tracer is when it's attached to a bulky molecule, 110 kD antibody, its rotational mobility is hindered, so it cannot tumble as efficiently as it would when it's free. So the polarization is very high, and this is at the start of the reaction. So you would get a number, something like 270 or something polarization units. Now, as the reaction progresses and as ADP is formed, the antibody has a much higher affinity for ADP, so it changes the partners. So the tracer is displaced off, and the ADP binds to the antibody here. And now you have the free tracer, which tumbles. So tumbles, meaning it keeps rotating, so it changes the plane of polarization. And now the polarization decreases. So this is a typical example what you would see. You would start with a high polarization value, and as even if you increase the enzyme concentration or the reaction proceeds farther, you would see that the polarization drops. So not very intuitive, but it's a negative readout. So you get a curve that's as a product is formed, you get the polarization decrease. Now, some people don't like to see that, so we plot it as delta MP, which kind of is the same result, shows delta MP. Now, we have a fluorescent intensity assay in which we took the exact same antibody and we conjugated it to a quencher here, okay? So when it's bound to the tracer, the quencher will reduce the fluorescence or quench the fluorescence of the attached tracer molecule. So you start your reaction with low fluorescence or very basal level of fluorescence, like shown here. And as the reaction proceeds, as you start accumulating ADP, the ADP, since it has a higher affinity, would bind to the antibody, displacing the tracer, and you now see increase in fluorescence, as shown in this example. It's the PI3 kinase, and you can see that there is an increase in fluorescence as the ADP accumulates. The third kind of assay flavor that we have is the TR-FRET readout. And in this, the antibody is attached to the lanthanide terbium. And because of the proximity of terbium and the tracer, there is a high FRET. So there is a transfer of energy when you excite the terbium. It gets excited, transfers its energy to the tracer, and as the reaction progresses, again, same principle, the antibody binds to ADP, displacing the tracer away, and the FRET is removed. So once again, the FP and the TR-FRET, though they perform very similar between the three, the FP and the TR-FRET gives you a negative readout, the FI gives you a positive readout. But the principle of the assay, the reagents for the most part remain the same. They are all direct assay. And now what is again the advantage of using a direct assay? I did talk about, we use a far red tracer that minimizes any interference from organic compounds. B, we don't have we don't have any enzyme other than the kinase. So you don't have to go back and deconvolute. You can very easily weed out your hits from interfering compounds. And the way you do that, you always sometimes have colored compounds or highly fluorescent compounds in your library. And so you just look at the intensity and you would see like a 5-fold increase in intensity, and you can weed them out very simply that way. You don't have to go back and run any deconvolution assays like you would have to do if you are using a coupled assay system. But the greater, I I would think the biggest advantage I see as a scientist in using this direct detection is that you could use this in real-time mode or kinetic mode. So as the ADP is being formed, you can the antibody would bind to it, and it sometimes you would see when you work with not kinases but ATPases, there's a lot of product induced feedback loop inhibition. As ADP accumulates, it could inhibit, and the antibody basically just pulls it down, and it works really well in that mode as a kinetic assay, as a real-time assay to measure the ADP. And this particular application helps us measure not just the IC50 values or the potency of the compound, but also parameters like residence time or the K-off rate. So how long is the inhibitor bound to the target, to the kinase? So things like that helps, medchem progress at a very fast stage, and you can do it parallelly. Like you can do the potency as well as the K-off values to make sure your SAR takes into account both these parameters. SPR is a good method to use, but it's very expensive, and these are just very, very cheap reagents that you could easily get IC50 and the K-off rate. So I talked about the affinity of the antibody for ADP versus ATP. So we have 140-fold affinity for ADP over ATP. So what that means is the antibody loves ADP, but it also binds to ATP to a certain degree. So in the order that the antibody likes ADP, then ATP, then AMP. And we really do not understand why it likes triphosphate more than monophosphate, but that's how the affinity of the antibody ranks. But this just means that because the antibody does bind to ATP to a certain extent, we will need to adjust the antibody concentration depending upon your initial ATP concentration. So for example, if you're using 1 micromolar ATP, you will be okay using about say 0.5 to 1 microgram per ml antibody. But if you use more, like say 1 millimolar, you might have to increase the concentration of antibody. And we make that very simple by having like a easy to use linear equation. So you just pop in the value of ATP that you plan to use, and the equation would give you how much antibody you would use. So that's one of the ways to optimize for antibody. As we mentioned, the assay is really simple, mix and read assay format. Here is an example. At Bellbrook, we primarily run all our assays in low volume 384 well plate. And when I mean there are so many different kinds of 384 well plate, our assays requires low binding or non-binding plates. And the reason is, unlike ELISA assay, we don't want the antibody to get very comfortable and sit down coating the plate. We want it to be, if you can imagine, free floating in that well, so it can capture the ADP that's formed in your kinase reaction. A typical low volume plate that we recommend has a total volume of 30 microliters, and we run the assay in a final reaction volume of 20 microliter. And and here is an example where we show that you run your enzyme reaction in 10 microliter followed by detection reagents. So you run your enzyme reaction depending on your target. So 1 hour or maybe 2 hours if it's a really low turnover enzyme. And then you add your tracer antibody and a stop reagent that we also include in our kits. The stop reagent is nothing but EDTA containing buffer. Most kinases require magnesium, so the EDTA would chelate the magnesium, thus quenching the reaction. Now, as I mentioned, you could run the assay in real-time mode where you just add your tracer and antibody without that EDTA containing buffer, and you can read the plate every 30 minutes or 15 minutes to get the kinetic data. So we do say that you run it in, because most of the data generated in-house is in low volume 384 well plate, but you could use the assay in a 96 well plate. You could also do it in a 1536 format. In a 1536 format, you can split it as 3+3 or even 4+4 reactions in 96 well plate. We recommend using half area 96 well plate, so you split it as 25 plus 25 just to minimize the cost of reagents. And I want to just add one more thing here before we move on. Even though we kind of do this, we are saying 10 plus 10 or 5 plus 5, we give the user complete flexibility to design the experiment as per your needs. Say, for example, you have a kinase that's really going very slowly and you want to increase the amount of ADP that's produced or make it more sensitive. You can run the system as 15 plus 5 or even 18 plus 2. It does not matter. You just have to, keep the amount of tracer, like whatever concentration of tracer, 2 nanomolar or 4 nanomolar in 20 microliter reaction and the amount of antibody in 20. That's all that matters. The user has the flexibility to sort of design the assay whichever way they want. We just propose an example here. So talking about sensitivity, our assay is pretty robust, and Z prime is a measure of how reproducible and robust the assay is. So as I said, shown here are 3 different formats: the FP, the FI, and the TR-FRET assay. And you see so many different standard curves here. So, this is 1 millimolar ATP to ADP conversion, 100, 10, 1, and 0.1. I'm just going to, for convenience sake, just focus on 10 micromolar ATP and ADP standard curve. And what we mean is when you say 0% conversion, we mean that it has only 10 micromolar ATP and no ADP. And 100% conversion, therefore, would mean that all the ATP has been converted to ADP. So it has 10 micromolar ADP and no ATP. Now, that's a good thing because it gives you the ceiling and the bottom of the curve, but you're more interested in initial velocity conditions, which is around, less than 30% conversion. So shown here is a 10% conversion, meaning 1 micromolar ADP plus 9 micromolar ATP. Okay, so that's kind of where your kinase would be converting at the initial velocity condition. And you can see it's really robust Z prime here for FP, FI, and TR-FRET, and they all perform very similarly. And even if you go lower, you see the same thing. The reagents are very stable. This is a 21-day reagent stability. We don't want you to try this out. This was just putting our reagents through, keeping them, the tracer and the antibody at 37 degrees. And this is just done with the FP assay reagents because it's just not conjugated to anything. It's in its native form. And you can see they're right on top of each other, really rugged reagents. And this is a plate signal. So you add all the reagents and come back the next day and read the plate, and still the results are right on top of each other. So pretty rugged signal, both reagent and the plate signal. So now So now I'm going to move into kinases and innate immunity. So inflammation is a very conserved host reaction that is initiated whenever there is trauma or tissue damage or infection. So that leads to changes in tissue homeostasis, blood flow, immune cell activation, migration, etc. The end result being secretion of these cytokines or interleukins that happens. And there has been so much of progress made in the last couple of decades in identifying these signal components. And you can see how many kinases, all these blue ovals in these figures that was borrowed from this Nature review, indicates the kinases that are involved in the innate immunity pathways. So it needs no further saying that, the pharma companies are so interested in identifying, validating, and developing small molecule kinase inhibitors as a very new, generation of anti-inflammatory drugs, including feedback loops, inhibitor specificity, and combination therapy. So, it's been very, very well pursued targets. So in general, whenever bacteria or viruses or other constituents of bacteria or viruses like lipopolysaccharides are recognized by high affinity receptors, what happens is there is a lot of oligomerization of adapter proteins, and it leads to activation of multiple kinases, the blue ovals that we showed in the previous figure. And those are crucial modulators of the innate immune response. The cytokine production is an essential part of the immune response, and the kinases involved in regulating the production of master cytokines such as the tumor necrosis factor or the IL-1 have been one of the most attractive targets for anti-inflammatory therapy. And so there has been a lot of interest, and what Bellbrook has done is we have taken some of these kinases in the pathway and have shown as that you could use transcreener assays to study these targets or to identify inhibitors. So as the first example, we will take AMP kinase as our example. And AMP kinase actually does not need much introduction in the metabolic homeostasis of cells. It's kind of an old enzyme in that scenario, that it's been studied a lot for homeostasis of cells. But more recent studies, especially after the coronavirus hit us, it's been SARS-CoV-2 uses, as has been published and studied in the last 2 years, uses the ACE2 receptor to infect the human body. And research has shown that AMP kinase can increase the expression and stability of the ACE2 receptor. So once the coronavirus enters the body, then it leads to downregulation of ACE2. So there has been a lot of interest in AMP kinase to study the SARS-CoV-2 in that context. But this paper that in JBC that came out recently also shows that AMP signaling is very important in terms of the STING and interferon beta expression. And we have, in this image that I borrowed from that paper, it shows that AMPK signaling can, include by chelation of intracellular calcium, as shown here, or by, just removal of AMP signaling by developing small molecule inhibitors. And it also inhibits the STING-dependent signaling that by affecting the interferon beta regulation. So how do you do assay development for any of these kinases? The first step would be to identify the optimal ADP2 antibody concentration. Now, I did mention in the beginning of the presentation that we have a linear equation that you could use, and you, in fact, could use that. That's a wonderful starting point. But sometimes if you choose to use a protein, a full-length protein, or something that might, just be sticky and bind to the tracer or the antibody, we kind of want you to determine the optimal antibody concentration by titrating it. The second step would be to identify how much enzyme you should use, and that enzyme concentration should give you a good window and also give you a good initial velocity condition. And the third one is to, validate it for HTS conditions, like can you run a screen, can you do dose response curves, etc., etc. So we'll go over these examples one by one. So the first step is to optimize the antibody concentration. So we pick a buffer, and usually this step is, you always, like for a kinase, you require magnesium chloride and a tris-based buffer. We add a detergent here, typically at 0.01%, and we use bridge, but it works with Triton too. You could use very low concentration of any detergent, and the detergent is just to prevent non-specific binding. But your target might require some additional cofactors, because there are some kinases that might require calcium or manganese. So a literature search would be very helpful to finding the most optimal buffer. But this is typically where we start with when we don't add additional things if you don't need to. So when you run a binding curve, you get a nice sigmoidal curve. And we typically pick the EC85 concentration, which is where the graph starts going down, if you may, where it starts going down. So you pick any antibody concentration in this area. What it means is when you pick something very low here, say I pick this concentration, your window is very less. You only get like 100 MP maximal window. If you picked somewhere over here, you're using a lot of antibody, you may not be that sensitive. So you pick the best compromises somewhere in this area. So we call it the 85% concentration, where you get a good compromise, you get a good window, and you also get a good sensitivity. So we pick that concentration. So the conditions we used was, okay, this buffer, we added ATP. So when you run a binding curve, you do have to include everything but your enzyme. So you have your ATP, which we used at 25 micromolar. And this again, you do a literature search, find what the KM is. And in or around the KM is a good starting point to develop your assay. And substrate, for this particular kinase, we picked SAM's peptide at 20 micromolar. So when we did that, we kind of got a concentration of about 15.5 microgram per ml. Now, this is not too different if you had used the equation, but say if I had used your full length protein, this would definitely be shifted. Now, second experiment that you would run is, therefore, you would use that enzyme, the antibody concentration. Now you would do a enzyme titration. Now, typically, when you're starting out, you do like a kinetic runs, but this example is an endpoint assay. So what you see here, this nice thick blue curve represents the complete reaction. When I say complete reaction, it means it has both ATP, which is the donor nucleotide, and the acceptor peptide, SAM's peptide. So this is a complete reaction. Now you need to run the controls, and you see that in the absence of ATP, it's really clean. And in the presence of just ATP, no peptide, you see something at the highest concentration. Now, I'll show you another example where it's more pronounced than this, but it definitely, the non-productive hydrolysis that happens without the peptide is a very common phenomenon seen with kinases. And most of you who have worked with kinases, have seen this before. When you have just ATP and no peptide, what happens is sometimes water gets into the binding pocket and you would see non-productive hydrolysis. So it's good to run all these controls. Now, our assay by general, it's not just Transcreener assay. Any competitive binding assay by nature is going to be nonlinear. So you will get a sigmoidal curve when you plot the raw data. So MP or RFUs or AFI, whichever assay, you will get a non-linear curve. So it's sigmoidal plot. Now, how do you know? Like usually people think like this is my this is a very small dynamic range. It's very hard to decide based on the raw data. So we ask you to run a standard curve under similar conditions. So in this case, the standard curve would be 25 micromolar ATP to ADP conversion in the presence of the SAM's peptide. So when you run that, you can interpolate all these values that you're getting here into the product formed. And here you can see the scale is different. So we are only looking at, around 0.5 microgram. So it is this lower end, which definitely doesn't look very linear in the raw data graph. But when you convert that into product formed, you can clearly see that the contribution due to non-productive hydrolysis, in this case, with just ATP, is it's minimal. There is barely any contribution, and you pick a concentration that is at the initial velocity condition, meaning the linear zone, and you could go even higher. As you can see, you would lose the linearity only around the highest concentration, meaning like at 2 or something. So until 20% conversion, you would still be here. 2.5 represents 10% conversion. So you can go as high as 4 micromolar ADP production, and you'd still be linear here. And so what I'm trying to say is, how do you decide what is the best enzyme concentration? There are two factors that you should look at. One is if you pick an enzyme concentration, say I'm picking 0.1 nanogram per microliter, see if you get a good shift. You need to get at least 100 MP shift. And sometimes if you don't want to use that much enzyme, you can go longer. You can run a 2-hour reaction, just so you are making more product if you want to conserve on the enzyme. So the first rule of thumb is to make sure you get a good window. Are you getting at least a 75 to 100 MP shift? The second rule is to run a standard curve and see at that concentration that I'm planning to pick, is it linear? And of course it is. If I'm picking anywhere between this range, I'm making about 2.5, which is less than 10% conversion. It's linear and it's also giving me a good window. So those are the 2 rules of thumb that we observe as we pick the optimal enzyme concentration. Now, the third thing is, okay, you did validate that, okay, there was very minimal contribution due to non-productive hydrolysis, but all this, does that translate into a good parameter to run a screen? So to do a screen, we could take, there are several ways to validate this. You could do plus or minus enzyme, plus or minus substrate, or just do it with an inhibitor. But the point being, we just run a Z prime experiment and show that with the enzyme, you still get a very robust, reproducible Z prime that shows that you could run a good window. And what you're trying to establish here is, if you need like a unambiguous, when you run a screen, you want to make sure that you can identify the hit unambiguously. You call out something and say, okay, this is definitely a hit. And then you could proceed to test known inhibitors, and the staurosporine is like a broad inhibitor that's used, as a broad kinase inhibitor. So here we just show a titration, an IC50 curve with 2.1 nanomolar IC50. So these are just the couple steps that you will have to follow. I mean, it doesn't, it's like, the first thing you do antibody, and then you find out your enzyme concentration, and then you run a dose response curve and a Z prime, and you're ready to do your screen or run several other dose response curves. So that's the simple assay development step that we propose here. So this is the next case study that we will show today. It's IKK beta. It's another kinase that was shown in one of those oval boxes. So the IKK complex consists of 2 kinase subunits. One is the IKK alpha, IKK beta, and the regulatory subunit called NEMO, which is the NF-kappa B essential modulator. So what happens is, when the CGAS pathway is what we study a lot here. So when cytoplasmic DNA is inside the cell, it activates the enzyme CGAS, which goes and phosphorylates STING, activates STING, which oligomerizes, gets phosphorylated, and phosphorylates another kinase, TBK1, which has also been validated at Bellbrook Lab. But a series of, multi-subunit pathways, but it ends up phosphorylating IKK alpha, phosphorylates IKK beta, and which goes and phosphorylates this. So NF-kappa B is kept in it itself is a heterodimer, and it's kept sequestered in the cytoplasm by binding to the inhibitor of kappa B protein, or I-kappa B protein. So what happens is, when IKK alpha and IKK beta gets phosphorylated, they phosphorylate this protein, which gets ubiquitinated and is degraded, thereby releasing this NF-kappa B to go and do its job, which is activate the transcriptional program. So what we did here is, again, we pretty much use the same buffer system for IKK beta, and here we picked ATP as 10 micromolar, and the substrate was IKK-tide, and we usually buy our substrates from AMPK or some other peptide provider. And you can see, again, do a binding curve with everything but the enzyme, and pick a EC85 concentration, which is close to 15 microgram per ml. Now, when you run the when you try to optimize enzyme concentration, you can see that it's not as clean as what you saw in the previous example of AMP kinase. So here is your complete reaction with ATP and IKK tied. But when you have just ATP, you still see a very, there is a lot of non-productive ATP hydrolysis happening even in the absence of IKK tied. Now, this is something you would encounter a lot when you work with kinases, and that's why I picked this as an example so I can show that, it's not always clean like how you see in AMP kinase. And when you convert your data, you can see that, yes, it's minimized, but you still see that it is, there is a significant production of ADP even in the absence of the acceptor substrate. Now, there are two ways you could try to, one is pick a concentration where you have minimal amount of I would probably pick somewhere in this window. So it's linear and the contribution by non-productive hydrolysis is completely, it's not eliminated, but it's minimized. Okay. The second thing is to increase the concentration of your peptide substrate. So typically when we start with 10 micromolar or something, but when you see a curve like this, you could try using 25 micromolar or something, increase it twice to see if that would further, increase the complete reaction, shift it to the right, whereas, keep the other reaction where it is. So those are the two ways you could circumvent this this issue that you would certainly encounter with kinases. So because it gave us, you do see that we kind of run Z prime with all the controls here. So you can see that, even in the absence of substrate, you would see a sort of a signal here, but you get a good Z prime nonetheless. And you can see the staurosporine titration. And if you look at some of our competitors' data, you would see that those controls are never ever shown in the absence of the peptide, or even in the absence of, they only show one curve that looks really great, but you have to really tear them or like dissect them open to see what is happening even in the absence of substrate. So we kind of show all our controls so you can get a feel for it, like what kind of response you would get. What else can you do with these assay development? Sometimes, like I said, you could titrate the IKK tide, and this is what we ended up doing because we ended up using much higher concentration instead of 10 micromolar, which gives you a good reaction, but you could use higher IKK tide. Or you can titrate also your ATP. Now, this is a pseudo KM, I call it, because it's not actually a KM response. To do an actual KM, you have to pick one enzyme concentration, various different time points, run standard curve. And we have done that. And if you want to refer to a method how to do that, please visit our website. We have application notes that go over exactly how to set those reactions up. But this is like a quick and dirty way to sort of see if you're using the right ATP concentration, or can you go any higher. So you can titrate your ATP in a quick fashion, and so you can titrate your substrate IKK tide as well. And then you could also do it in other formats. Sometimes, so this is where we use slightly higher IKK tide concentration, but you can see that now the shift is, slightly better than what we had seen with the FP format. So you kind of, the way I do it is I like FP a lot. So I develop the assay. I kind of get all my initial conditions using the FP format, and then repeat with those, the final conditions in FPI and TR-FRET. So you can see, when you increase the IKK tide concentration to about 30, 25 micromolar, you minimize the amount of non-productive hydrolysis, and you separate it slightly more than what you had previously seen. And the other thing I wanted to point out is, we typically, if you see our website, everybody wonders like why Bellbrook always shows their data in FP format. It's just because I think when we developed this assay like 20 some years ago, I think we only had FP, and everyone is like comfortable with a certain assay, and so when most of the scientists here like the FP, so we run everything in the FP mode. But that doesn't mean that the FI and the TR fret are not good or doesn't work. So this is an example where we took several inhibitors and compared the IC50 that we obtained from FP versus the TR fret versus the FI, and you can see that they're very, very similar. There's a good correlation between all three assay formats. So the choice of the detection format is only your personal preference and the instrument that you have, and all instruments are not made the same. Some are. I know the instrument vendors may not be very happy with me saying this, but some instruments are better than others, and especially for some readouts. And if I were to give an example, I would say if you're using, say, TCAN M1000, it's a spectacular instrument. It's a monochromator. You can tune any wavelength. It's great. And it works great for FP and FI, but not so good for TR fret. And if you were to pick like, some other instrument, FP would be spectacular, like Envision. FP works really great in that. So every assay, and TR fret too. So every plate reader has like, one assay that it does so well. So you do have to, your personal preference, the instrument availability, that sort of should be guiding you which assay to choose. The assay themselves, they perform really well and give kind of very similar Z prime. The FP is just more rugged in some ways because it is the antibody is in its native conformation. Nothing is attached to it, so it's a little bit more rugged than FI or TR fret, which is not a big surprise because in a TR fret you are attaching a terbium, in FI you're attaching a quencher, so you've got to be a little bit more gentler with those reagents than FP. But by far they perform just the same. So the choice of the detection format should be with what you're most familiar with and the instrument that's available to you. We have validated, I would say, a lot more kinases and even other targets, and shown are some example here. This is JAK1, shown here. You can see all the 4 things that we have done. And this is TBK1, another very prominent kinase in the innate immunity pathway. So visit our website. There are a lot more enzymes, not just kinases. There are also helicases, some ATPases, those are all validated. So check out that. Yeah, I think we have a free app note. So yeah, if you have not yet downloaded that, please go get your download visit our website. I think it has a lot of additional information that we didn't cover today, or more data that you could take a look at. And although this was more, tailored for kinases, we also have other assays. Our Transcreener UDP assays, Transcreener GDP assays. This is for GTPases, GAPs and GAPs. This is for glycosyl transferases. We have AMPGMP assay, which has a whole slew of applications and targets that you could use them for. And then we have some assays we have 2 assays for epigen, like methyltransferases. One is based on the Transcreener assay called the Epigen assay. The other one is aptamer assay, which is 2 pieces of RNA that comes together when SAH is formed. We also have adenosine assay, which is, a coupled assay. You should check that out too. And I think one of the newer ones is the C-gas assay, which detects the C-GAMP, cyclic GAMP assay, and this is also available. I think most of them are available in more than one format. FP, TR-FRET, etc. So check that out if you have any interest. And the targets listed here, you would find most of those data in our website. And if you don't, just reach out to me, Justin, or anyone basically at Bellbrook Labs, and we would guide you to it. With that, and please, like, this is our ordering info. If you want to order any of the product kinase assays, just go to info@bellbrooklabs.com. Send us information. Our website has a lot of interesting information. Check out our blogs, etc. And if you have any questions from today's talk, shoot any of us an email in any of these. With that, yeah, questions.
Yeah, so one question that we've seen a little bit here is, I'm, as an example, this research is interested in a certain kinase target. Is there a way of determining if it will work with the assay?
Yes, definitely. So the first thing I would say is go to our website. Most often you would find something, or check out the literature to see if anyone has published anything with Transcreener assay. But in general, any kinase that ours is a universal assay. So basically, any kinase should work. We may not have validated some kinases, but that doesn't mean it wouldn't work, because most often it's just very hard for us to get a good quality enzyme. The protein is the most difficult source, to source it out. So we may not have data just because of that. But you could always, try an assay, try it out. We have some smaller kit size available, and if you send us an email, we would also help you, suggest, okay, try this peptide, try this vendor, etc. Since it's a universal assay and we measure the product formed, most often any kinase should work. And just follow these 4 steps, like antibody titration, enzyme titration, and then proceed to other things.
Sure. Here's another question for you, Mira. Do you use very high quality ATP to avoid the presence of any ADP?
That's a very, very good question. Yes, I mean, so that we get our ATP actually from Sigma, and I would say if all the reagents, I would say the most careful I would be is with ATP to not to not dip into it all the time, because if you have any nuclease contamination, it would break it down. The ATP that we supply with the kit is one of the high quality ATP. It's about 99% pure. But again, that 1% impurity could have some ADP. And that is one of the reasons we always optimize our antibody concentration to sort of, get an idea, a feel for it. It's very important to make fresh buffers. And everybody has their way to do it. The way that I have been doing it is I make my buffer, I aliquot them and put them away in -20, and I thaw like 1 out. So, that you're using the same buffer through your prep, and it's not contaminated with, and always use nuclease-free water and don't double dip like everything else, I guess.
Another question, Mira. You mentioned the assay can be run in kinetic mode. Have you ever seen any assay interference by the antibody, tracer, or detection reagents?
Yes, I have actually seen some interference in some cases, and I think that was there was, I think, one particular target, and I cannot remember on top of my head, but I would think it was one of the HSP proteins that I tested a while ago that showed some interference. It showed completely different reagents results when I ran it in kinetic mode versus endpoint. And I've run several kinases and ATPases, and that was the only target that I had seen some kind of interference with the detection reagents in the kinetic mode. And we really didn't understand what was happening, why it would work in kinetic mode in a different way versus an endpoint method.
All right, it looks like we have time for one more question. This was pretty general, but are there any potential tips you have for running the Transcreener assay like this? Any common questions that come up often?
Yes, one, I would say it's several, but for the lack of time, I would say definitely a couple things that I would touch upon is um, the plate is very important. Sometimes I have had users run them in any kind of 384 well plate, and that's why I stressed a little longer when I was talking about the plate. It has to be non-binding, low binding, and sometimes these it says low binding 384 well plate low volume, but the low volume is sort of a very loose term because if you go to any plate supplier, for example, Corning or Perkin-Elmer, they have plates with 100 microliter volume, which is also called low volume, and a 40 microliter or a 30 microliter volume that is also called low volume. So if you take a 100 microliter plate, volume plate, and that's the capacity of the plate, and you're running the assay in 20 microliter volume, you would see a lot of light scattering happening because of that, the high capacity, high volume plates. So I would always say check what is the total capacity of the plate. And if you just have that available to you, then you will have to increase your volume, not to 20, but to, increase it to 40 or 50, something like that. That's one question I always we talk about plates a lot here. The other thing is buffer. It is very important to use nuclease-free water when you make buffers, etc. And with kinases, like especially when you start working, I think especially with TR-FRET or FI reagents, I would say the terbium component of it is a little bit more finicky and might be more, it's more, what is the right word to use here? It's a little bit, it depends on the environment it is, if you may. Like, for example, if you have heavy metals and like say some kinase for some reason, if you have like, gosh, what is it? Iron or something. I'm just throwing something in there. It might interfere with terbium a little bit. So just pay attention to what is in the buffer. So the environment does matter. And this is more important for the TR-FRET assay. And this is a property of it, the lanthanide. So you have to just, see what is the cofactor, and that should be the deciding guide. The second thing is they don't like to be freeze-thawed multiple times. So keep it, we say 5 times, can go as many 6 to 7 times, but please, don't like just leave them at 4 degrees. And, the FI and the TR-FRET reagents are a little bit more finickier than the FP. So just gentle handling. I would say aliquot always. And with ATP, nuclease-free water always helps.
Thank you, Mira. I think that's all the time we have for today. And we have, with any other questions, please contact us via email, info@bellbrooklabs.com. Someone will definitely follow up with you. I think there's a couple more questions here, so we'll try to reach out to you and answer those as well. So a copy of this webinar will be sent within the next 24 hours. We'll also send you a copy of that associated app note that Mira showed before, and it provides tons more details on assay conditions for a lot of different targets, and that should be very helpful. So with that, thank you for joining us, and goodbye.
Thank you.